Within this activity, we will learn all about the genetic code. Then you'll have a chance to crack your own codes and test yourself on what you've learned. Our genetics provide our cells with a huge amount of information. The information tells our cells what they should become, how they should replicate and repair, how they should grow, and how they should function properly. It is our genetic information which determines various inherited characteristics, including physical characteristics such as eye colour. So to put that a little bit more simply, our genetic information acts a little bit like a recipe book. Since following different instructions leads to very different outcomes. Cells store genetic information in DNA molecules, which are located in a part of the cell known as the nucleus. In order to fit the huge amount of DNA into the cell's nucleus, the DNA is wound up very tightly to form an X-shaped structure, which is known as a chromosome. The region of DNA that encodes the genetic information is called a gene, and there are lots of different genes. The genes act as instructions to make molecules called proteins, which are essential for our bodies to grow and function properly. So, what exactly are proteins? Well, they are larger molecules made up from a chain of smaller molecules called amino acids. The chains then fold to form proteins of a certain shape and size. The correct folding of a protein is very important for it to function correctly. Proteins have many different functions in our body. Some are structural and influence the shape of our cells. Some are hormones, which regulate various different activities within the body, while other proteins are involved in the body's defense system, such as protecting our body from germs. So, put a bit more simply, proteins are made from a string of molecules called amino acids, which are a bit like building blocks such as Lego. Therefore, the size and the shape of the structure built depends on which blocks are used in which order. For example, using the same bricks put together in a different order can result in a very different outcome. So, how exactly does DNA code for these proteins? DNA is composed from basic building blocks, which are known as bases. These bases are referred to by scientists as A, G, C, and T. Therefore, a simpler way to think of the information provided by DNA is as a long sequence of the letters A, G, C, and T. In a similar way that letters of the alphabet have to occur in a certain order to make words or sentences with a particular meaning, the order of the letters A, C, G, and T in the DNA sequence is particularly important for determining which amino acid building blocks are used to make a protein. As I previously mentioned, the DNA sequence is located in the cell's nucleus. This is not the site where proteins are made. Proteins are actually made at a different site called a ribosome. Therefore, the information provided by the DNA sequence in the nucleus needs to be converted into a form which can then travel to the site of protein production. This is done by creating what is called a messenger RNA, which is able to leave the nucleus. Messenger RNA could be thought of as a little bit like a message in a bottle or even an email, where its purpose is to get information from one location to another. The process in which DNA is converted to messenger RNA and then finally to protein is together called the central dogma of molecular biology. The conversion of the DNA into the messenger sequence is called transcription, while the decoding of the messenger sequence to work out which amino acids are needed to form the protein is called translation. So first, let's explore transcription. DNA normally occurs in two strands, with the bases located opposite from each other being complementary. So this means that A is always found with T and G is always found with C. During the process of generating the messenger RNA, one strand of DNA acts as a template. One of the differences between the DNA code and the RNA code is that the RNA code doesn't have any T's, and instead the T's are replaced by U's. If you have a pen and paper handy, I wonder if you can use the template DNA code provided and the conversion information in the table to work out what the sequence of the messenger RNA could be. the answer. If you didn't get it right, don't worry. So the T's in the DNA template equal A in the messenger sequence. The A's in the DNA template equal U in the messenger sequence. C in the DNA template equals G in the messenger sequence, while G in the DNA template equals C in the messenger sequence. So now we understand how the DNA code is converted into a messenger sequence. Next, we will explore translation. Translation is the process of decoding the messenger sequence to identify which building blocks are required to build the protein. So, it's the messenger sequence which tells our cells how to build a protein. Every 3 letters in the messenger sequence is read like a word in a sentence. Each word tells the cell which amino acid to use next in the protein chain. Scientists call the 3-letter word a codon. Scientists have a table which lets them work out which amino acid is coded for by the 3-letter word codon. In a similar way, I wonder if you can use the table provided to work out what shapes the messenger sequence is coding for. The first letter of the codon can be found along the left-hand side, the second letter can be found along the top, and the third letter can be found along the right-hand side. So just for an example, AUG would code for a rainbow. Feel free to pause the activity if you need longer to work this out. Here is the answers. Don't worry if you didn't get it right though. We're now going to explore what happens if mistakes happen during the process. Changes that occur in the DNA sequence are known as mutations. Here is an example of a mutation that is called a substitution mutation. This is when one letter in the sequence is swapped for another. One of the G's in the DNA sequence has been swapped for a C, which is highlighted in yellow. This causes a change in the messenger RNA sequence and results in a change in the codon. The new codon sequence codes for a lightning symbol. A change like this, which changes the amino acid sequence, will likely influence a protein's function. This is another example of a substitution mutation. This time, a T in the DNA sequence has been swapped for a G. Again, this causes a change in the codon sequence. However, from the amino acid table, we can see that the new codon still codes for the original shape. Mutations like this, which do not change the amino acid, are called silent mutations, as they will not change the protein function. Other types of mutations that can occur include the addition of an extra letter, which is known as an insertion, or the removal of a letter, which is known as a deletion. These insertions or deletions cause a shift in the sequence, which is called a frame shift. This is an example of the addition of an extra A into the DNA sequence. It shows that a frame shift caused by a mutation can cause a change in many amino acids. These mutations can cause problems for a protein's function. Pathology is the study of the origin and nature of disease. One way to further understand disease is to study a patient's DNA sequence in order to see if any mutations can be identified. Sometimes the identification of mutations can help with diagnosis, which is a process of determining if a patient has a certain disease. Meanwhile, some mutations can influence how a disease may behave. Thank you for taking part in Cracking the Genetic Code. I hope you've learnt something. If you want to test your new knowledge, then please answer some of the following questions. Now that you've completed Cracking the Genetic Code, I wonder if you can answer these questions. Can you remember the name of the building blocks which together make up proteins? The answer is that proteins are made from amino acids. We have learnt that a codon is made from 3 letters which represents bases. But what is a codon? A codon is the 3-letter sequence which determines which building blocks or amino acids is used to make a protein. What are changes in the genetic sequence called, and do these influence the protein's function? The changes in the genetic sequence are known as mutations, and if the mutation causes a change in an amino acid, then they can change the protein function.